crack on. We have a next exciting talk, which is from Vineet Tiruvadi, and he is a MD PhD student at the School of Medicine, Emory, and Georgia Tech. And he is going to talk about network oscillations in antidepressant deep brain stimulation. So over to you, Tiruvadi.
Great. Thank you so much. Thank you for the introduction. I'm Vineet. I'm excited to present today on network oscillations and antidepressant deep brain stimulation. Did you all see the slide just change?
Yes, I think, but we do have another slide on top.
Do you have the notes on top?
Yeah.
Okay. Let me try it one more time. Sorry about that. I thought I selected just the window. Do you still see the notes?
It's coming. Yes, we still see the notes.
So
we can't see your slide. I mean, you could have, yeah, no, that's better. So we have your slides.
Can you see the slide right now?
Yes, now we can see that.
Okay, that's fine. I have another computer over here. All right. So thanks again. Sorry for the technical difficulties. In the next 12 minutes, I'll review DBS, its use in depression, and the reverse engineering paradigm we're using to study it. I'll talk a little bit about the major results from our empirical work, most of the work that I did during my PhD, before discussing the biophysical modeling efforts and future directions for the field. Okay. First, the work is a result of a huge team effort, and I want to thank everyone that helped, especially the support from Whitaker Foundation and Emory MSTP, and a special thank you to the patients who ended up being crucial collaborators in the effort. So, deep brain stimulation is a neuromodulation therapy involving the implantation of electrodes into specific parts of the brain. In this picture, that's implanted into the subthalamic nucleus. This enables a neurostimulator to deliver therapeutic patterns of electrical currents, typically around 130 Hertz, into brain targets associated with disease. Implanted DBS leads have multiple electrodes available, with at least one electrode being near a desired target and the stimulation voltage being a parameter that can actually be used to adjust how much electricity is being delivered to engage that target. Now, DBS is commonly used for motor disorders, but promising applications in psychiatric illnesses are growing, with multiple brain regions and multiple disorders being studied. Today, I'm going to focus on subcallosal cingulate, brain target, and depression. Depression is a psychiatric disorder characterized by persistent sadness, sleep difficulties, suicidality, and other non-specific symptoms. Neuroimaging studies have actually helped identify a network of brain regions associated with depression. One node in particular, the subcallosal cingulate CG25, it plays a central role in depression, being metabolically hyperactive in depression. Metabolically hyperactive in depression and hypoactive when successfully treated. So the picture above and below show how SCC changes its metabolic activity as a result of depression state. Now, DBS in the SCC showed early efficacy in treating depression, but recent advances and refinements in our lab have actually specifically implicated white matter tracks, the axons in the brain, as the necessary therapeutic targets, specifically for white matter tracks, the uncinate fasciculus, the forceps minor, the cingulum bundle, and the thalamic fibers. Here you can see each one colored in a different color. Now, this was a retrospective study. In a subsequent prospective study where individual tractography was targeted in each patient, individualized, we demonstrated an improvement in the efficacy, which implicates a little bit the subcallosal cingulate white matter is actually the therapeutic target we need to hit. Now, this SCC white matter DBS is what we currently do. We actually specifically target white matter tracks and not the broad gray matter of the SCC. So this is essentially what our group performs today, and we're continuing to study it. Now, how do we study it? Because while SCC white matter DBS appears to work in treating depression, its mechanism of action remains unclear. Modern efforts in DBS are moving towards adaptive strategies that rely on measurements and algorithms instead of physician assessment to optimize DBS stimulation. The clinical devices that have been developed give a unique opportunity to study how DBS changes brain activity and how brain activity is linked to behavior. Now, using a prototype DBS device capable of both stimulating and recording in patients, in this case, the Activa PC+S, which came out originally in 2013, we can actually measure local field potentials multiple times a day over years in our patients that we're treating. The results I'm presenting today are from 6 patients implanted with the PC+S between 2013 and 2018 who have all successfully recovered from depression. Because of the clinical nature of this work, we had to rely on several different approaches to study our therapy, including benchtop testing of the PC+S, intraoperative recordings, extraoperative local field potentials multiple times a day, and monthly experiments with combined LFP and EEG. Now, because of the small sample size, we had to take a different approach, one built on models that the clinicians already use to treat patients, and rely on the data that we're able to collect, sparse and noisy though it may be, to refine those models that the clinicians are already using. So our first goal, as every good first goal should be, is characterizing our measurement. How good are our measurements? And in particular, our PC+S recording had this issue called mismatch compression. And we had to correct for that before we could actually analyze the brain oscillations we cared about. Then we wanted to study how the brain oscillations actually related to behavior to identify a depression readout. Can we actually predict how depressed a patient is from the oscillations we can record? And then finally, we wanted to study how DBS modulates the physiology of the brain. And this gets at characterizing the specific network action of on-target SCC white matter DBS instead of broader SCC regional stimulation. I won't talk much about mismatch compression in this talk, but suffice it to say we developed a pre-processing strategy to remove the impedance mismatch caused artifacts in our chronic SCC LFP recordings. And this enabled us to actually reliably study oscillations in the brain. So I'll focus mostly on 1 and 2. Diving into 1. So to identify SCC oscillations related to depression, we implemented our therapy exactly how we implemented it before, which is implanting the patients, turning the stimulation on 1 month after the implantation, and then following them weekly to assess how depressed they are. These are the Hamilton Depression Rating Scales, our way of measuring depression over the course of about 8 months. And higher corresponds to worse depression. In parallel, and blinded to the clinicians, I analyzed LFP recordings that were measured approximately once every 4 hours over the course of 7 months. And these we downloaded every week and enabled us to actually have LFP recordings, multiple LFP recordings a day, and about 70, 80 recordings a week associated with each of these weekly Hamilton Depression Rating Scales. This data set was split into a training and a testing set, about a 60-40 split, with each recording associated with that week's depression score. An elastic net regularized linear regression model is trained through a nested cross-validation approach before it's assessed in the 40% training set. And the 40% training set performance, it ended up being approximately 10% of the variance we found in our empirical Hamilton was accounted for in our oscillations. And we find that the model actually identified specific oscillations as the most important, specifically theta and beta. And interestingly enough, there was a hemispheric sign difference in their contribution to the depression readout. Now, importantly, this model is robust to the inclusion of stimulation artifacts. These are the artifacts caused by mismatch compression over the course of 7 months, and including that as a feature doesn't change the results too much. We found that SCC LFP oscillations could actually be used to predict a part of the depression signal in all of this work. And we're working on putting this stuff out, and the code should be available. The next piece of it is the network action. So to characterize this, we combined LFP and EEG recordings and stimulated at 2 different regions. One on target, which is where the SCC white matter target for each patient is, and then one off target, which is either 1.5 millimeters above or 1.5 millimeters below. What we did is we took recordings before stimulation and during stimulation and found the response, the change during the stimulation from the pre-stimulation baseline, and used these segments. We segmented them into 2-second segments and used these segments to characterize both LFP response and EEG response. And interestingly enough, in the LFP, we saw under the on-target condition, on-target is blue here, we saw no major change in either the left SCC-LFP or the right SCC-LFP oscillations across all low frequency oscillatory bands. Surprisingly, when we're off target, when we're actually not stimulating the therapeutic target, we see broad increases in activity, specifically in alpha and beta. Now, this is a little bit counterintuitive until we realize that we're actually trying to stimulate white matter tracts. In each patient, we're going to the white matter tracts and we're specifically targeting those. So when we look at the EEG under the similar exact same experiment, so we were collecting EEG at the same time, when we look at the white matter tractography associated with being on target, we see that we mostly hit right cingulum bundle in our patients. And that right cingulum bundle hit is manifest in the EEG by a right-sided alpha increase near the parietal tracts and a temporal decrease. Now, comparing this to off-target stimulation in each patient, we see a very, very different response. These are scaled together. And off-target evokes a very, very different response in the EEG, suggesting we're engaging a very different group of white matter tracts. Actually makes sense. Now, the last thing we did was actually do a classifier on just the EEG segments and found that we were able to achieve approximately a 90% classification accuracy in differentiating between on-target and off-target stimulation using only 2-second segments. And the channels that ended up being the most important, this was using an L1 regularized SVM, we found that the channels that were most important were, as expected with the tractography, this right-sided EEG channels that correspond with the cingulum bundle and left-sided temporal channels that correspond with the left-sided unsniffed fasciculus. And this work is being done in a similar way. I am running out of time, so I'm going to try to, I'll skip through the summary. I think that was fairly clear. We see local changes in the SCC. We only in the long term, not in the short term. In the short term, we see specific network activity in remote regions measured by the EEG. And this gives us a clear idea of what the mechanism of action of SCC white matter DBS is. Now, the modeling work done here is we actually introduced the virtual brain, a dynamical systems platform, a whole brain modeling platform, into the middle of our tractography and our EEG. And what we were able to do is predict where our primary EEG channel changes would be, and then predict where our secondary EEG channels would be by applying the connectivity matrix of the virtual brain parcellations, and then using that as a mask on our empirical EEG measured in each patient. And what we ultimately found was that the primary channels were mostly increases in alpha, and our secondary channels were mostly decreases in alpha. Now, further work needs to be done to validate this in our other patients and at different stimulation voltages, as well as to actually include dynamical neural mass models into the middle using the virtual brain. So in summary, we have DBS of SCC white matter alleviate symptoms of depression and actually evoke specific changes to network level oscillations that we're able to observe at multiple scales for the first time. Oscillations in the SCC LFP can actually be used to partially decode depression state, but obviously other brain regions are involved with depression, so we need to find a way to expand this readout. And careful analysis of LFP data is needed to actually confidently say you're analyzing brain oscillations. Clinical data is noisy and sparse, so actually taking a data-driven approach is challenging. Instead, I would suggest doing a data-congruent approach where you take models that clinicians use and ensure that they're consistent with whatever data you're able to collect. And finally, this reverse engineering style approach may be the way we can actually find the long-term mechanism of antidepressant DBS, and tools like the virtual brain and connectivity matrices and connectivity analyses are actually going to be crucial to actually dissect what's happening in our patients. So finally, thank you. Feel free to follow me on Twitter and read a little bit more about the work at my website. Any questions?
Okay, that was great. Yeah, thank you. Thanks a lot. So we have a couple of questions coming in for you. So one question was actually typed in the chat box, so I will just quickly ask that. Are all patients receiving bilateral DBS?
Yes. So when in the experiments I showed today with chronic recordings, yes, chronic therapy is bilateral. And in the acute recordings, the LFP and EEG recordings, they are bilateral stimulation.
Okay. Okay. We have another question coming from Paul, such that Paul, I'm trying to make. Would you like to ask questions yourself? I can make you unmute and you can ask your question yourself.
Sure. Can you hear me?
Yes, we can hear you.
Yeah, very nice talk. Do you see any longitudinal changes in the EEG patterns as a function of whether the stimulation is on target or off target?
It's a great question. So I personally didn't do any of that analysis. Some folks in our group are exactly doing that. We've done the same experiment, the impulse response style experiment with on target and off target at 3 time points, at 0 months, 3 months, and 6 months. I don't know what that data looks like. And the challenge there is our EEG is going to be our signal is very small, and we're going to be sampling it very sparsely. So I don't know the answer to that.
Okay. Thank you.
Thank you. Thanks a lot. Okay, so